Spent 6 months doing that.
Thanks everyone for coming. I know I told people last lecture that there'd be nothing, you'd only be responsible for the last 3 lectures on the test. So if you want to leave, now's your chance to get up and go. You will learn stuff, some new stuff, and there will be some review that will help you on the test. Now you saw the, what, anything here?
Any of the stuff in here.
There's nothing new here that will be on the test. Okay. But there may be some hints and other things. I'll review a few things. They may be on the exam. So if you did the homework, and I also sent out some review questions, that's going to be the format of the midterm. There should be no surprises. It's going to be, you're going to do a lump parameter model. You're going to have to derive a few things. Huxley model, I won't ask you to derive much, but I'm going to ask you a bunch of questions that kind of probe whether or not you understand it. And then, also I should say, I don't try to complicate things. If something seems really complicated, take a deep breath, think about it. Often I kind of put things together so you might logically get from one to the other. If your TAs can't help you, also I should say, sometimes I make mistakes. So if there's something on the review questions or homework that just doesn't make sense to you, send me an email, and I'll get to you. I'll call or something. So I usually make myself available the night before the final. So if you have last minute questions, or sitting there and can't figure out something, send me an email. Okay, I guess that's it. So let's get started here. I kind of zipped through these sections, so I wanted to just maybe do it again and sort of rehash a few points. The heart is this flow-chambered organ that pumps blood. There's a specialized conduction system, but most of the ventricular myocytes of the muscle, they're all activated. The important thing is they're all activated on every heartbeat. So, unlike skeletal muscle, where we can recruit more or fewer fibers, we can pick different types of fibers to modulate the force. Here, you can't modulate the force using recruiting different numbers of cardiac muscle cells. You get them all. Here on this slide, there's not really too many points to make here. One thing, as I was saying, this gives you a good idea that heart cells, this is the cell membrane, this is a connection between cells. There's gap junctions, so excitation can pass cell to cell. But you can see these things are packed full of myofilaments. So the main thing in a heart cell is myofilaments. And then you have transverse tubules. Again, these are where the outside can dip into the cell. So inside the T-tubule is really extracellular space, and they don't connect with other things. So T-tubules are not continuous with, say, sarcoplasmic reticulum. And recall, sarcoplasmic reticulum is this container that holds calcium. And so if you look at things like a frog or an amphibian, it doesn't contain a lot of sarcoplasmic reticulum. So this seems to be like a secondary system that evolved to help the system run faster. So something like a frog has a relatively slow heartbeat, whereas something like a mouse, as you're saying, beats 600 beats per minute and per second. Hummingbirds, these things can go very fast. So the sarcoplasmic reticulum is sort of a new mechanism. And in fact, you don't even see it in fetal tissue, so it seems to develop even later within, say, humans. There's two kinds of connections, phosphoadherins and macroadherins, and these are here to mechanically connect the cells. As you can imagine, you had this array of discrete heart cells, and they're all contracting. You're not going to get the organ to contract unless there's a solid connection. So these are two of the connections that I want you to know.
What's the difference between those two?
That's a good question because I always forget. So I wrote it here. So phosphoadherins connect the actin. So really you can think about this as connecting, one cell is going to probably end on a Z line, and then another cell butts up against it, there's another Z line, and you want to connect those mechanically. So this is saying these anchor actin, which is thin filaments. Now there's also macula adherens, and these are sometimes called desmosomes, and they connect intermediate filaments, which is another cytoskeletal element. So as I was saying, we used to think of cells as kind of a bag of water, but now we realize there's this whole cytoskeleton. And one of the important components is intermediate filaments. So there's really actin, which here in the muscle we know is the thin filament, but within other cells it's just a structural protein. That's one that nature seems to have reused. And then another important component is intermediate filaments. You see those in the smooth muscle slides. And then there's also microtubules, which are important for the muscle actually. So this is a picture of what we call excitation contraction coupling. So it's the cell that excites, electrical activation. You get a whole cascade of events which ends up in the contraction of the myofilaments. And so it's saying this slide was made by a guy that loves calcium handling, and he made these very small, but you can see the real muscle cell. This is what I'm interested in modeling. This is really what the cell's full of, are these guys, myofilaments. But anyway, for this diagram, so what happens when the cell excites, you get calcium through these voltage-gated channels. These are called L-type channels, often, but all you need to know really here is it's a voltage-gated calcium channel. So calcium comes here. This is the primary way that calcium comes in. And when it comes in, it causes a secondary release from this structure. So this is sarcoplasmic reticulum. Again, it's a storehouse of calcium. So when this comes in, you get a secondary release. And interestingly, about probably 10 times more calcium comes out of sarcoplasmic reticulum as comes across the cell membrane. And so this is a specialized system. What happens, well, I'll tell you a little here. But again, this seems to be an adaptation to allow the system to run very quickly. So instead of moving all this calcium in and out across the cell membrane, you have this secondary thing. So once it comes in, then you pop it out with a bunch of calcium, which can flood the cell, activate the myofilaments. Again, you need a lot of calcium because this is the end effector here, which is the cell that's full of these myofilaments. So it really takes a lot of calcium to activate the system. So another secondary way that calcium comes in is through this sodium-calcium exchanger. So 3 sodiums, it exchanges 3 sodiums for 1 calcium, and it turns out this guy ends up being voltage sensitive. So when the X potential and the voltage goes up, it actually runs in what is sort of the reverse mode, bringing in calcium. Now here, this is showing another voltage-gated calcium channel. This guy and this guy are actually the same. My pointer's going to die early today. So these two are the same. Just the issue is this one is facing this release channel. The release channel is, by the way, called a ryanodine receptor, and it's actually shown here. It's a very huge channel. But some of them, they just put calcium into this bulk cytoplasm. This is probably like 90%, 95%. There's a few of them that are scattered around that calcium. So basically, these 3 influx mechanisms go, increase cytoplasmic calcium level, activate myofilaments. And now what's important is, okay, now that we've activated it, how do we shut down the system? So calcium can go back into sarcoplasmic reticulum through something called a circle pump. And circle pumps use ATP, so it's using energy to actively pump that back in. So again, this is a specialized system. So the cell's using ATP to sort of make this run fast. Now some calcium goes back out across the cell membrane. Some of it through the sodium-calcium exchanger. So during the rest periods between beats, the sodium-calcium exchanger runs in what's called forward mode. So it extrudes calcium by letting in sodium. And then there's also this thing called the sarcolemmal calcium pump. And so this uses ATP to extrude calcium. Now, say in a human, probably about 70% goes back into sarcoplasmic reticulum, and about 30% goes out across the cell membrane. And by far the largest is sodium-calcium exchange. This one here is sort of a slow trickle out of the cell. Now, just one second. So the slow trickle, is important because you can imagine, over time, even a small current integrated becomes important over 10 or 100 or 1,000 beats. So even though there's maybe a small net flux, over time is sort of important. Is there a question?
So the main difference here between the skeletal cell is that the L-type channels don't go right into the rhinogate receptors.
Yeah, random receptors. So yeah, so that's an important distinction. Thanks for bringing it up.
So what happens here is more of a calcium-induced calcium release, as it says. And so then for the other one, they call it voltage only because calcium goes straight in to the.
No, actually,
how does that work?
Yeah, thanks for bringing that up. So here, right, so in cardiac, an important distinction is calcium comes in, and then this channel is also calcium sensitive, and it causes a secondary release. So this is called calcium-induced calcium release. Now in skeletal muscle, there's this foot process that actually connects this channel with this release channel, and there it's voltage sensitive. So that's a voltage-induced calcium release. And it doesn't actually require the calcium going through the channel as much as the voltage, the voltage change during action potential. And so, this kind of gets at one of the themes here is that skeletal muscle is sort of more all or none like a neuron. Every action potential neuron is exactly the same, but in skeletal muscle, every twitch won't be exactly the same, but it's much more like all or none. So in skeletal muscle, once the membrane activates, it causes this secondary release of calcium. In cardiac muscle, calcium comes in and causes a secondary release. And this is more about how you can modulate cardiac muscle. Because again, in skeletal, you can trigger it with a neuron. You have a motor unit that activates the muscle fiber. Everything's sort of all or none. Here in the heart, you get all of the cells, and so there's other kind of modulatory mechanisms. This is showing the T-tubule. Again, it's extracellular phase of the T-tubule.
I had a question about how the calcium goes back out of the cell afterwards. You said 70% goes back into the sarcoplasmic reticulum, and then 30% is pumped out of the cell. Is it only pumped or through the exchanger? That it's not like there are channels? Like, I guess that wouldn't work because of the concentration difference. So there are no channels bringing calcium out.
Yes.
Only the pump and...
Yeah, so this circopump, so here, circoprotein. So the amount that goes back in depends a little on the species. So something like a human or a dog is probably about 70% cycles back in. In a mouse or, say, a rat where things are going really fast, probably about 90% goes back in the sarcoplasmic reticulum. So it's a little bit species dependent. And then, what doesn't go back in the sarcoplasmic reticulum then goes out across the cell membrane. When I say, it's not completely imbalanced. And then here you have this NCX is this sodium-calcium exchanger. So that's one way it gets out, and that's the majority of extrusion. So that lets in sodium down its concentration gradient and pulls calcium out against its concentration gradient. And I should say, this is the same in all the cell types. Basically, this extrusion mechanism, so either circle pump or things going on across the cell membrane, it's the same mechanism. So if you happen to get asked in a question about whether they all have the same extrusion mechanisms, that they are. So look at all the slides and try to see that it's consistent. This is just a simple diagram that sort of breaks these pieces up. It's not too much new here that isn't explained before. This is just maybe a couple pieces. There are actually calcium buffers. As I was saying, you have to really get a lot of calcium in your cell to cause this calcium transient to activate the muscle. Later in the action potential, you have calcium coming in through these voltage-gated calcium channels. And probably most of that's actually going into sarcoplasmic reticulum. Not for this beat, but for the next beat. And so, again, this is cardiac, where there's different modulatory mechanisms. Let me see, and I'm also saying that there is this kind of funny thing that happens is while calcium is coming in and getting released from sarcoplasmic reticulum, it's actually being pumped back in at the same time. And there's no way to sort of turn these things on and off. And someone actually asked about channels. I mean, these exchangers in this calcium pump, are these channels, or they're sort of light channels, but I mean, in the sense that they're protein structures, they create a pore through the membrane. But for this pump, it uses ATP to move the charge against its concentration gradient. Here, sodium calcium exchanger is like a channel, but it lets in 3 sodiums for each calcium that it extrudes. So this is showing action potentials. So this is one for a rat. So showing an action potential. You guys did voltage clamps, right? That was part of last section. So this is just showing either action potential or a voltage clamp with step response. And this is showing rat versus rabbit. So rat's very fast. It's not quite as fast as a neuron spike, but it's fairly quick. And again, it's running at about 10 beats per second, so you really have to go up and down pretty quickly. Rabbit is very close to human. It's, say, about 200 or 300 milliseconds. But the point here really is that whenever the cell's depolarized, whenever it's at a high voltage, you actually have calcium coming in. And probably the important thing is that first bit of calcium that comes in, this is the calcium current here, triggers the release. The next ensuing long bit of calcium coming in loads up sarcoplasmic reticulum to the next bit. So, I think it's the same here. Unfortunately, these things aren't so simple. When you look at calcium coming in, it has different roles. One role is to trigger release. Another role is to load sarcoplasmic reticulum for the next beat. And it also contributes to the shape of the action potential. So again, unfortunately, physiology is a little bit complicated at times. And, we can't say this has one single role. Actually, things get overloaded. What this is showing is these are twitches. So this is in cardiac muscle. And so if you pace at a steady frequency, you get sort of a fixed, it comes to a steady state. But here we're going to accelerate the rate. As we accelerate the rate, it actually goes up to a higher force level. And so, again, I've said intuitively, we know when our heart races, you can feel it contracting much more vigorously. So one way we can modulate the output of cardiac cells is by speeding up the pacing. That brings in more calcium over time, loads up sarcoplasmic reticulum, and you get more calcium release. So here when we slow back, this is slowing back down, you get a funny transient, which you don't have to understand, but then you go back to a lower level. So, as I was saying, cardiac muscle is more like all or none. It doesn't matter how fast you pace it, you get sort of the same thing. And heart, it seems clear that part of the modulation.
Cardiac and then skeletal.
I'm sorry.
Yeah.
Yes. Skeletal muscle is more all or none. Cardiac, it's clear that part of the way you modulate it is by speeding up the heart rate. So another way that we can modulate it is by using hormones and neurotransmitters like adrenaline, and you get excited, fight or flight response, your heart contracts more vigorously. So smooth muscle, again, this surrounds our hollow organs, except for the heart. The important thing about skeletal muscle is, if you look per unit, so for cross-sectional area, it can contract with about the same force as the other muscle types. It's just intrinsically much, much slower. So there's a couple of types, single unit and multi-unit. I'm going to start to go things a little quicker. Here you have, these are, in the red are these intermediate filaments. So you get these dense bodies here which connect, these are really equivalent to like the sarcomere in a way. You have thick filaments in the middle, and then you have sort of thin filament like actin, and when the myosin turns on, it ratchets and sort of pulls this structure in. And so smooth muscle isn't arranged in the very regular sarcomeres, it's arranged in this more complicated structure. But overall, again, cross-sectional area-wise, when you try to normalize it, you can get about as much force out of smooth muscle. The gap junctions here, the gap junctions, there are channels that allow ions to pass and also other small molecules, so it allows for electrical and chemical communication. Phosphorylation, so this is a very common thing you see in cells for signaling. You have a kinase which will attach this phosphate group to a target protein, or maybe on a serine or a threonine. And then once you attach this, it changes the function of target proteins. And then you have a phosphatase that will then take this off and then return it to a sort of other state. And so, again, as you always think about anything you turn on, it's not valuable unless you can turn it off. People always think about kinases, but there has to be a phosphatase for every kinase. And actually, usually there's a few missing that I've discovered, but they have to be there. So, the important thing about smooth muscle, is that we have many different ways to activate it. So you can have sort of the same kind of potential-dependent calcium channel, which is the same thing as a voltage-gated calcium channel. So calcium may come in directly because the cell membrane depolarizes. There's also channels that may let calcium in because a neurotransmitter or hormone is bound. So this would also let calcium come in. Calcium can come in through the sodium-calcium exchanger at high potentials, where it runs in reverse mode. And there's also this kind of channel, which is a neurotransmitter or hormone binds. There's coupled to something called a G-coupled receptor. It activates this phospholipase C. And then really what's critical, which may show up on the test, is that it's going to turn PIP2 into IP3, and then IP3 travels off to the sarcoplasmic reticulum. Now, there's something called an IP3 receptor, and this is actually the same, more or less the same channel as the ryanodine receptor. So it's one of these big channels that lets calcium out of sarcoplasmic reticulum. So, just like skeletal is this, voltage-induced calcium release, cardiac is calcium-induced calcium release. What's unique in smooth muscle is you get IP3-induced calcium release. But they're very similar in that the calcium comes out of this reservoir called sarcoplasmic reticulum. Now, what happens, so all these different sources of calcium will give you a calcium transient. And then when the calcium transient happens, calcium binds to calmodulin here, and then calcium and calmodulin activate this myosin kinase, which is going to activate the cross bridges. And then to turn the system off, calcium has to go somewhere, so either it gets pumped back across the cell membrane or it goes back in sarcoplasmic reticulum. So I drew this arrow, which isn't in the textbook, but it should go back into sarcoplasmic reticulum. Some of the calcium coming into the cell will go back into sarcoplasmic reticulum, and some calcium will go out across the cell membrane, the calcium exchanger or this sarcolemmal calcium pump. Again, sarcolemmal calcium pump, sodium calcium exchanger, and circle pump. These are the three mechanisms which calcium goes out of the cytoplasm to turn off the system. So the primary way to activate muscle and smooth muscle is that a kinase directly activates myosin. So when calcium binds to calmodulin, it goes on to activate this myosin light chain kinase. That phosphorylates something called the light chain on myosin, and then the myosin will start to ratchet and do its thing. And then there's this, again, kinases and phosphatases work in this antagonistic pair. So there's also dephosphorylation from myosin light chain kinase. So essentially, once this guy turns off, then things go back this direction because of the phosphatase. This is just showing a picture. So if we look at what myosin looks like, there's really two heads together, the tail regions wrapped together. So there's two heavy chains, but there's also these light chains. And then the light chains, there's one of them called a regulatory, and this is the one that's phosphorylated. There's also one called central. But for our purposes, regulatory light chain is phosphorylated in smooth muscle, and it's the primary means to modulate it. In the other muscle types, assume it doesn't happen, and it's not important.
Oh, so you're saying in smooth muscle, the calcium doesn't bind to troponin? It actually affects the myosin and activates the myosin by activating the
Right. So in the striated muscle, skeletal, and cardiac, it binds to these thin filament proteins. And so it's not sort of indirect. Here, it actually binds to the myosin itself and regulates the speed of cycling. So, again, calcium can come in different ways. It can come through different membrane channels, but it can also there's also this way a ligand can activate a G protein, turning PIP2 into IP3, causing this release from sarcoplasmic reticulum. This graph is stripped down. There's not all the mechanisms, but I'm sure you get the idea. So IP3 causes this IP3-induced release from sarcoplasmic reticulum.
I have another question. So why doesn't the calcium just cause the release from the sarcoplasmic reticulum like it did in the cardiac muscle? Why is it different? Why is it different?
Yeah, so I think the difference is uranium receptors in cardiac muscle are somewhat different from these IP3 receptors. And IP3 receptors, which sit on sarcoplasmic reticulum, are sensitive to IP3. Now, not to confuse people for the test, but the reality is these channels are also sensitive to calcium, but it's just not as critical. So what I want you to know is in smooth muscle, IP3 is sort of the important.
And the IP3 actually comes from a hormonal?
Yeah, so the hormone and neurotransmitter say would bind to this receptor. There's sort of a second messenger G protein which activates something called phospholipase C, which converts that. So, but this is again sort of trimmed down and not show all the pieces. But yeah, so IP3 causes a secondary release of calcium. Calcium binds
in
to.
this case, it's like IP3 induced calcium,
Yeah.
but not.
Again, there may be a component of calcium-induced calcium release, but wipe that from your memory. It won't be on the test. If something is on the test, it would be IP3. Okay? So, calcium binds to calmodulin. So, and then calcium calmodulin activates this kinase, which then phosphorylates the channel. So this has this PI, that means that we put this phosphate group on the regulatory light chain. Once we do that, now we can begin a cycle. And the important thing here is that smooth muscle, you speed up the rate, it's going to generate force, it's going to generate shortening, but then you can lower down the phosphorylation level and stop the cross-reducing cycling. So they are what's called a latch state or a latch bridge. So it's like you shorten, but then once you stop cycling, you're actually holding on. It's generating force. And this is sort of our smooth muscles, for instance, in our capillaries and in our gut. You may want to hold the force without shortening. And so, just saying in the skeletal muscles, if I'm pushing on this, which would be sort of isometric condition, my muscles aren't shortening. But I'm using ATP, I'm generating force, but I'm using ATP. From the Huxley model, we know even when velocity is 0, we have cross bridges coming on and off. So we're using ATP. So what smooth muscle can do in a way is much smarter energetically, is that we sort of generate the force and then we stop the cross bridges from cycling. Now we still have the force. We're not doing any net work, but we're still going to keep the force. So, that's really, sort of the important take-home message I wanted to get to you about this. And then here, this is showing, force-velocity relationships. Again, these are sort of hyperbolic shape. It doesn't asymptote on the axes. It asymptotes either below or to the left. So this is showing a fast skeletal. This is showing slow skeletal. This is showing smooth. And so here, the stress really is force or tension. And you can see they all end up at the same place. And this is going to be force per unit area, or cross-sectional area. But you see that smooth muscle here is just a very small fraction of the shortening velocity. And then this is showing percent phosphorylation, so how much if we activated it. And then this is showing that V0, where the unloaded shortening velocity grows sort of linearly with phosphorylation level. And then we're showing that stress here, force, it's not linear, it's curvilinear, but you can see how you change phosphorylation level, you can modulate force. And then this is showing a family of traces for force-velocity relationship. So recall that for a Huxley model where we can assume skeletal muscles fully activated, we only got one curve for force velocity. But here where we can modulate crossbridge cycling, we then can get different curves. So one point is, I don't know if I brought out, but this sort of, the shortening velocity is very dependent on the ATPase rate. So something like insect flight muscle has a really fast ATPase rate. And so that allows that muscle to shorten very quickly. In sort of larger, mammals and such, it's slower, but, say rat is faster than human. And so a high crossbridge cycling rate is good because it allows you to shorten quickly. But the price you pay, which is a skeletal muscle, again, I'm pushing against something, I'm not doing that work, but because I have an ATP cycling rate, I'm using ATP. So this is sort of the trade-off. High ATPase rates can give you faster shortening velocity, but they also drive up the cost when you're saying isometric conditions and not doing work. Okay, so let's go on to thin filament activation. So, we touched on this before about how calcium binds to troponin and tropomyosin. This was the picture. This is sort of a nice, sort of very simple model here. Calcium binds to troponin. Troponin works with another protein called tropomyosin, which is this longer one. And when calcium binds, it causes an allosteric shift to expose binding sites. And now myosin can get in and bind to generate force. This we looked at before. This is showing, how these proteins sort of wrap around the two-stranded helix of actin in the thin filament. Actually, this was the one where he said, okay, if you look at low calcium, it's in this position. At high calcium, it's in another position where it exposes the binding site. Now, what happens at intermediate levels of calcium? We'll go on, but, from a macroscopic view of the world, you'd expect it maybe to sort of slowly shift. But in the microscopic world, things don't tend to work like that. It's going to be sort of flipping on and off, much like channels switch state. And these are sort of the same slides that we looked at before. This last time did not work. And I'm going to guess it's not going to work this time, but we'll give it a try. Oh, maybe it will work. Okay, so calcium comes bouncing along, it binds to troponin C, causes a shift in troponin I, which shifts troponin T in tropomyosin, and now it exposes this binding site, and now myosin can get in and do its thing. So this correctly shows that the ATP is turned to ADP and PI early, and then the head rotates, and then you need another ATP to come along to cause the cross bridge to detach. So this kind of quickly playing the game before about how does this differ from reality. So anybody want to throw out a few things? Any thoughts? Actin is a little longer. Yes, actin should extend. But what do we know? When a single crossbridge binds and rotates, do we get a net translation between actin and thick and thin filament? No, I mean, it doesn't. This isn't a Huxley. You have a lot pulling, again, hand over hand, a lot of things working in parallel. So this particular view is not really right. I mean, the reason that it's drawn that way is because it's simple. And to do it right, you have to sort of, the head would rotate and stretch that tail region. But that's hard to animate, so it's simple. A couple other things that are different here is that, how many myosin heads do we have together? 2. We don't know how they work together, so for simplicity, we draw 1. And then again, everything in this happens forward. Calcium binds, this shifts, that moves, this bonds. And in the microscopic world, everything's going to run forward and reverse. And in a net way, it moves forward. But it may have intermediate steps that go backwards and forward. So this is something new. So this is showing a forced calcium relationship. So you can do this in skeletal or cardiac muscle. It's a way where we fix the calcium level here, and we look at a steady state response. And if you did this, and you measured force, this is normalized, but you can just say between 0 and max. It looks like this. It's sigmoidal, but it turns out that it's fairly steep. We can fit it with something called a Hill function, which we'll get to in a minute. So, from what we understand, we sort of build a very simple model. We would say, okay, maybe calcium binds to troponin. So this is shown what's called a functional unit. It's a piece of thin filament with its regulatory proteins, which are troponin and tropomyosin. And here's a nearby cross bridge. So we're going to bind calcium to the troponin and tropomyosin, and then it shifts, and then a cross bridge binds. So this would be like a very simple state model, much like you have the channel state model. So you bind calcium in this step, and from here you have a couple rates which turn on the system. So what we call is non-permissive. So when these regulatory proteins block the cross-bridge binding, we call them in a non-permissive conformation. When they become permissive, we draw that sort of shifted, and now the cross-bridge is combined. So if we construct this kind of model, and so this is reasonable because we know troponin has one binding site for calcium, so one calcium ion binds. What you would get is a sigmoidal function that looks like this. Now, this is just sort of classic enzyme kinetic. If you look at any textbook about any kind of enzyme or any kind of single binding site, it's going to look like this. And so we can fit this with a Hill function. Hill function really only has 2 parameters. One is this CA50, which is the 50% value. So what concentration of calcium turns it on 50%? And then there's this is the exponent. Even though it's an exponent, we call it a coefficient. So this is the Hill coefficient. And so that sets how steep it is. If we were to plot log calcium versus log of force over 1 minus force, this is a way to take this curve of linear thing, straighten it out, and the slope will give you the Hill coefficient. And so what we find is the cardiac or skeletal muscle is much steeper than what you will get with a single binding site. So this is saying over a small range of calcium concentration, the system goes from fully off to fully on. And so this is important. Now, you get this later. I know you're going to get a few more sections on EC coupling and how heart cells work. But the basic idea is that we look at a calcium transient in a typical heartbeat. It changes by about a factor of 10. So this is 0.1 micromolar to about 1 micromolar. So this shift, this about 10 times change in calcium, translates to a much larger relative change in force. So for the heart to be an effective pump, what it needs to do is every heartbeat needs to contract. What it needs to between these relax. I mean, relax like almost turn completely off. Detach all the cross bridges. And this is highly critical to how the heart works. In fact, many diseases happen because not the heart can contract, but it can't relax. So to turn the system fully off, it evolved in such a way that, again, calcium doesn't change much, but force changes a lot. So when you take this relatively small change in calcium and sort of map it to a much larger change in force, it takes this nonlinear function. So I'm forcing you guys to think like an engineer here. You think about how to take a small change by turning into a big chain, this is nonlinear function. So this is that force calcium relationship, it's steep. And then, so if we're looking at heartbeat in a, to be more realistic, what happens in a heartbeat in a cardiac cell is the calcium transient comes up. And it comes up fairly quick after the action potential. After the action potential, you get calcium, there's calcium release. That drives up the calcium level in the cell pretty quickly. And as it's declining, the force comes on much later. So the peak force happens after the peak calcium. And so if one was to plot this steady state curve in the dash, during the course of a beat, it actually makes this phase loop. So calcium comes up very quickly, which is point A, actually let's look here. And then later, as calcium is coming back down, the force is coming up. So it actually makes this sort of loop around this steady state curve. So the system's actually set up in a way that's very steep, but it also operates sort of in a way that you can modulate force by how much calcium you release. That's kind of one of the critical things. Now, all this hill curves and stuff, it seems a little esoteric and maybe it doesn't seem so important, but actually if you look around biology, you see the same thing over and over again, and that is chemical reactions tend to not be very sensitive to concentration. So, often you will want something that's closer to like a threshold detector, or you want to switch. Below some threshold, you want it off. Above some threshold, you turn it on. So the other things like chemoreceptors for bacteria. There are other places where you want sensitivity, and then you have to have a higher Hill coefficient. So how do we get a higher Hill coefficient? Well, what you do is, we're going to talk about something called cooperativity, and that's when things, we're going to be kind of loose about it, but cooperativity here just means when proteins work together to bring about some kind of increase in sensitivity. So here we have the thin filament, again we have calcium binding component tropomyosin. So it's saying these tropomyosins connect together end to end. Each one spans about 7 units, but they work together end to end. And so what happens is they communicate. So when one turns on, it tends to pull its neighbor on. So this is part of it. So something called cooperativity type 1. This RU is for regulatory units. So one regulatory unit talks to its neighbor and helps to turn on the system. There's another piece of this, which is when cross bridges bind, they increase the affinity of troponin for calcium. So this is like a positive feedback mechanism. So calcium binds, we turn the system on, but once the cross bridges bind, then we even get stronger binding here. And as I was saying, this picture doesn't illustrate very well, but if you look at some of the earlier ones, the binding cross bridges actually move tropomyosin to a slightly different position. So it's sort of like as I was saying, it'd be wonderful if we think of calcium binding to the regulatory proteins as a switch that turns the system on and off. But there's this sort of interactiveness that once you begin to turn the system on, the cross bridges themselves turn the system on a little bit more. And in reality, what happens is cross bridges can hold the system turned on even after, say, this calcium is dissociated. Now, there's another type of cooperativity which is between the cross bridges themselves. So once you bind the first cross bridge, it may become easier to bind the second one. So this could happen a couple of ways. One is that there's this regulatory unit and regulatory unit communication. So maybe once you bind one cross bridge, it propagates down the line to the regulatory units. But there could be other mechanisms. For instance, this picture of the thin filament, these look kind of like beads on a necklace. The whole thing looks very stiff. But the reality is, these are protein complexes and they're a bit stretchy. So it could be that once you bind one cross bridge, you get some stretch which pulls, say, this binding site into better register with the cross bridge. So, the reality is there's these different cooperative mechanisms. They all may function to some extent. It's very hard, you know. We're still building lots of models to try to understand this. Go ahead.
What do you mean by more easily? Because don't they all require one ECP? Does
Well,
the process to be more easily like faster because they're aligned correctly, or?
Yeah, just like the Huxley model. Remember, you know, you have an A site and you have a myosin some distance from it. Well, you know, the myosin's here, and then somehow the A site gets pulled closer in the register. You'll get a
So it
faster.
kind of like lines them up.
It lines them up, yeah. So what I'm going to do, I think, is kind of zip through this section pretty quick. But I just want to introduce Monte Carlo methods, because you probably haven't heard this before, but this is, you know, part of what I want to do is introduce you to a lot of modeling techniques, because, you know, if you understand models and you're not, you know, scared away by them, they're actually a great way to encapsulate our knowledge, you know. You can read textbooks and understand, well, okay, this is what happens, but to really sort of understand it quantitatively, and really, you know, that's part of the really, you know, the beauty of this class is if you can begin to think about biology or physiology in a quantitative way, you're so much further ahead than other folks that just read the textbooks. I mean, they just, you know, they're not even the same league. So, just to sort of talk about Monte Carlo method. So what happens is, you know, when we have this kind of system like a channel, but this is done the same way. When you have calcium which binds to say troponin, you can think of this as on and off, like a channel opening and closing. So it's going to be reversible. We can have an on rate, in this case calcium dependent, and an off rate. And you know, instead of solving for like in the limit where you have a lot of them and you compute a scalar to give you the probability, what one can also do is just build a bunch of instantiations of this system. So we sort of build a lot and we make a transition based on generating a random number. So Monte Carlo, you know, so casinos, so, you know, you're rolling dice. You know. So here what happens is every time you roll the dice, you decide, well, do I switch states or do I stay in the same state? And then you have a probability which is set by the rates. So yeah, you know, I think, you know, if you're interested enough to read it, you can read some of the details. But that gives you the essence of it. If you build a bunch of instantiations of, say, a channel opening and closing, in this case it's calcium binding to regulatory proteins, or regulatory protein shifting, or cross-bridges binding, and then what you can do is to split time into discrete steps. So say a delta t, and at every delta t, you decide whether or not you stay in the same state or you switch out of that state. The probability is set by your rates, and we're going to draw random numbers. Now, if this was other years, I would have, there's a homework problem that shows you how this works. But, you know, same way with channels, you know, if you did a bunch of runs, it would be stochastic, but if you average enough, then you can get towards that steady state solution. So let's get this. So, you know, why is this important? So, you know, we built this, I talked about this very simple model where you went, this is sort of your functional unit, you have a cross bridge, you have a little bit of thin filament, you have a regulatory protein. This 0 means off, it means no calcium bound, and N is for non-permissive. You bind calcium here, and now this hasn't quite turned on yet, but here's the place where the system turns on, this shifts, cross bridges bind. You would also have this state over here, which is where cross bridges may still be bound or cycling, even though the calcium has disappeared. So this, you need this for thermodynamic consistency. Again, in the microscopic world, things, there's, you know, microscopic reversibility. So things that run forward should also run in reverse. And then, because we're using these Monte Carlo methods, what we can do is now couple the units together. So in this case, these rates here depend on the state of the neighbors. So what we're going to do is we're going to have this term here, gamma, And gamma has an exponent n. And so when n gets larger, it drives the reactions this way. When n gets smaller, it drives reactions that way. I know this is quick, but it's not on the test. So, but this is actually sort of, again, this is a system that's a lot of similarity to other systems. So again, muscle maybe seems a little esoteric, but this is, you find this kind of stuff around a bunch of places. But anyway, suppose we're a regulatory unit. We're gonna be the middle unit. Now we could have either both of our neighbors off, or one neighbor or the other neighbor on, or we could have both neighbors on. And then depending on how many neighbors, this sets this n value. And then going back a slide, sort of the n value drives the rates of this. So when you have channels, let's bring up, channels tend to be easier, or you've probably learned about channels. Each one is an independent system. And so its behavior, you can, when you look at the ensemble behavior, it sort of simplifies because each channel is acting independently of the others. This system's different because each unit depends on what its neighbors are doing. And that totally complicates things. It makes the math much harder. And so what happens is, this could be a piece of thin filament. We have all these regulatory units. They bind calcium and they turn it on and off. And if each of these would have 4 different states. It could be calcium bound or no calcium bound. It could be on or off. And so unfortunately, to do this, we have 4 to the 26 states. So now, if these were independent, then we'd have only 4 states. But because they're coupled for a whole thin filament, we have 4 to the 26 states, which is a huge number, which we can't compute using the old ordinary differential equations. So, trying to give you a little intuition here. So, if this is a piece of thin filament, and we're going to say, okay, this thin filament is 50% activated. So 50% of the units are on and 50% of the units are off. So one way you might do that is like this. Another way you could to do that would be to have this unit off, this unit on, this unit on, off, on. So alternating. Now, this gamma term, which is the communication between neighbors, is kind of like stretching a spring. So when you're different from your neighbors, it's like stretching a spring. So this is favorable because you're only stretching one spring. If you did this, you'd be stretching every spring. So the problem here is that this is energetically favorable. This isn't, but when you try to compute the behavior of the whole system, you have to include all of these. So you have to include the likely events, but even the unlikely ones. And so you have to use a Monte Carlo method. Computationally, it's very expensive because you have this huge system, you're generating random numbers.
What do the different positions set up in the battery?
Sure. I'm sorry. So this is permissive. So when they're on, it means if there's a cross bridge nearby, it can get in and bind. These guys are off, so they're preventing cross bridges. So again, both of these systems are half on and half off. But this is more likely to happen. It's energetically favorable. But when you try to compute the behavior of the whole system, you have to consider all of them. Now, one way we solved this was using something called icing model. This comes out of magnetism and magnetizing materials. So it turned out that we could take this approach for magnetic materials and map it to a biological system. So in a material, you have spins up or down, and they're going to be flipping because of thermal noise. But because they're magnetized, they tend to align. And if you apply a magnetic field, they tend to align one way or the other. But they're constantly flipping. And so you have to, again, solve this problem of how do you figure out the ensemble behavior? It gets very complicated when you have coupling. So we were able to solve that, and spin going back. And so we could figure out a model here shown by the solid line that very well fit the real data from muscle. In fact, this, to my knowledge, is the only model that really fits this and explains, can't say explains, but recapitulates the biology with a mechanistic way. Now, if we tried to do the same thing with the Monte Carlo model, you get a similar result, but it's very noisy. I don't remember, I mean, this is like 2 weeks of runtime on the server. Not a super fast server, but, you know, a reasonably fast server. So, in the Monte Carlo method, where you're sort of binding and unbinding things, you're tracking units, turning on and off, you're tracking cross bridges, binding and unbinding. In this case, they can bind and they can rotate. So, in that case, it's just very noisy. Even if you have a big computer and you run it for a long time. So Monte Carlo methods are nice because we can sort of map to the underlying biology, but they're extremely costly computationally. So, but you know, when you have these systems where you have proteins talking to each other, either regulatory proteins, or there's systems that you're going to hear about, I'm sure, with channels where, for instance, calcium comes in this voltage-gated calcium channel, and then there's a randomly receptor sitting next to it. When this guy opens, all of a sudden you have coupling. And that problem becomes something like this, where you get the Monte Carlo method. So why don't I end there with that? And so what we're going to do is, because we actually do this every year, people like it. So what we're going to do is, we're going to try to explain this in a more conceptual, intuitive model about how this system works. So, you know, what we want to sort of understand is, you know, why is this steep? Why is it sort of over a small range of calcium go from off to on? And then also, sort of how do cross bridges affect the system? So what I'm going to need is I'm going to need about, give me, say, I'd like six women to be part of this. I need women for this. You'll understand why in a second. So I know we've got a couple guinea pigs in the front. Anybody else? I know there's always in the class there's somebody who's done it. I'll start picking people. Okay, you in the orange.
You get extra credit.
Yes, in the orange. I know that you want to be part of it.
They need 6 more.
In fact, because you guys are co-located, I'm going to grab you too.
I think we need one more.
Then we need one more. One more female. Come here, you're looking. Okay. Okay. See if I can turn it off. Hold on. I don't know if I can turn this off or not. Okay, so I need you guys sort of lined up, side by side, spread out a few bit. Okay.
They're recording.
So you guys are going to be regulatory proteins. We don't have the thin filament. We have to sort of imagine the thin filament is here behind them. So you're going to be regulatory proteins. And as we taught, they can be either non-permissive off or permissive, which is on. So what I'm going to do is we're going to have you go like this. This is off. So go a little bit further apart. You guys are holding hands. I like this.
We are.
Okay, no, this is great. This is the first time.
I was not doing that.
I like the camaraderie here. We got them, you know, you guys are Viennese. You're all okay. Now hold hands again. That was good. Okay, good. So this is off. Now go like this. This is on. Okay, I got hold hands. I like this. Okay, we're going to practice. This is on. This is off. On. Off. Okay, so now if we were connected like this, if I turn her on, go ahead. Make her wave. Okay. I turn her off. Everybody go off together. Okay. See, in this way, they're all sort of talking together. They're, you know, when this unit turns on, it propagates down the line. So even though, each one binds one calcium. So what I'm going to do is I'm going to endow you with the calcium, which means you're going to turn on when I give you a calcium. Or, the reality is you don't turn on, you tend to turn on. Again, in the real, biochemical world, things may sort of, fluctuate, but you're going to tend to be more on. Now, for a thin filament, what I'm going to do is, you guys aren't connected hand to hand. Okay, you can let go now. But a more realistic model is that you're connected with an elastic link. So grab this. So this is why I need women, because if I did this with guys, someone's going to go, and someone's going to get injured. You guys are, I know, you guys might be just as, I got another one. Okay, there's one more. Okay, so now, what's going to happen is, when this unit turns on, it's going to want to pull the neighbors on, but it may not go on yet. So it's sort of like stretching the spring. Now, this is more energetically favorable because now the springs are shorter, but this is possible. So this is sort of the idea of this cooperativity type 1. It sort of tends to create uniformity. Okay, you can go down. Yeah, we need both arms at the same height. So, sort of to understand why this is important is that, like, I'm calcium here and calcium binds here. Okay, you got calcium. You're going to want to turn on, but your neighbors are going to try to sort of hold you down. So we'll let you stay down for a second. And now, I might find another calcium here. Okay. Now she's going to want to turn on, but maybe her neighbors are sort of holding her down. Now, what's going to happen is there's going to be a point where now we're maybe about halfway. And guess what? Now maybe we begin to turn on. Even though she doesn't have calcium. It tends to make everything sort of uniform. So we all turn on and off together. This is very nice. Okay, what we'll do is we'll turn you off for a second before I turn you back on. Okay. Now we need some guys. Give me a...
So does it have to be more than like 50% of them have calcium to get all of them turned on, like greater than 50%?
So does it...
To have the force to
So again,
control the others?
it's stochastic. It's sort of stochastic. So, less than 50%, you tend to stay off. Greater than 50%, you tend to turn on. But again, every different possible, there's a bunch of different possible combinations. And, again, to really calculate that, consider all of them. Okay, 3 guys, real quick. 3. Okay, you guys here. He's covered in space, so I will not call him. Okay, he was being shy. He covered his face, but he really wanted to get up here. Okay, great. These guys are the cross bridges. So what we're going to do is, these are myosin. So what we're going to do is, now we're going to turn back on our thin filament. So, again, you try to go on, but you're having trouble. Hold her down. Okay. Okay. You want to turn on? Yes. Okay. So now we're about 50% turned on. Okay, go ahead. We all flip up. Okay, you're a cross bridge. Just put you in here. This guy binds. Now it turns out the cross bridges, this thin filament stuff tends to be fast. It may take a while for the cross bridges to bind. So we bind the cross bridge. Okay, let's bind you too. Okay, and now you bind. Okay, so maybe this is about. Okay, I need you guys to like hold them up. So, I know you don't want to touch this guy probably, but. So now we're this is probably like peak force. We have as many cross bridges as we want. And what happens is, recall, at peak force, calcium may already be coming down. So what I'm going to do is I'm going to pull the calcium off. Now, she's going to want to come back down, but guess what? This cross bridge is blocking her. So pull yours off. And now you want to relax, but, oh, I'm sorry. That was not supposed to happen. It's hard, they can't relax. There's actually the myosin can physically hold that thin filament active. Now what's going to happen is, this guy may want to come out. Actually, you know what? I'll pull you out. But don't come down yet. Okay, I'll pull you out. Turns out, actually, probably maybe one cross bridge can hold that whole thin filament, which is about 26 units. So that one guy alone is holding everybody up. Now this is a problem. Well, that's a problem, but this is what happens: is cross bridges can cycle. So now I'm going to, take all the calcium off here, but you guys are all going to stay on for a minute. And what can happen is this: is that while you guys are on, even though you may not have much calcium bound, you get back in there. Now you come out. So just the sort of presence of cross bridges themselves to hold the system on. And then you go back in, and you come out. You see what's going on? So there can be sort of this regenerative nature of just having cross bridges. Now, when calcium comes down, the system has to eventually turn off. So what will happen? Okay, now you guys can shut off. So, this is kind of what we think happens in a muscle. So let's give them a round of applause for doing that. Okay, thanks. So we have just a couple minutes left to close out a few thoughts here. Okay. One other important point that we didn't get to talk about is that in cardiac muscle, when you go from a short length to a longer length, it becomes much more sensitive to calcium. So in the heart, you're going to learn about something called the Frank-Starling law. And basically what the Frank-Starling law says, however much blood you return to the heart, a healthy heart can pump. So, if we're walking around, we're not really doing a lot of work, we're not pumping a lot of blood, but if we begin to race up the stairs, all of a sudden, you need to generate a lot more circulation, move a lot more blood. And so what happens is, as you expand the heart, you sort of fill it with more blood between beats, it's got to contract much more strongly to eject the blood and sort of keep up with the demand. So one important thing about heart cells is the amount of stretch modulates the force. And so when we talk about how to modulate the force in cardiac muscle, there's this ability to pace it at a higher rate, you get more force. That basically brings in more calcium. We can also use hormones, but another important thing is using length. Now this won't be on the test, but this is for your knowledge that this is an important thing. And a lot of that comes about because of this cooperativity we just talked about. In fact, unfortunately, the mechanisms are still a little murky. Models keep coming out. We're all still wrestling this with a bit to figure out how it all works together. What we're going to do is skip this with the mechanism. By the way, this is a heart cell being stretched, and so what you can see is a striation, or a striated muscle. You can actually see the Z lines. I mean, this is the sarcomeres in here. And so one thought about how this works is that as you stretch the cell, because it's mainly water, it's isovolumetric. So if we make it longer, then it gets thinner, and it pulls the thick and thin filaments closer together. And that may modulate the attachment rates of cross bridges. So that's one mechanism. Let's get that. We think some of this length sensitivity may come about from titin, because titin, remember, is this protein that goes from Z line and actually runs down a thick filament. And so it's also elastic. So as you lengthen or shorten the cell, it's at a very good position, a perfect position to sense length. And there have been some experiments that have modified this genetically to take out some of the units and it changes the length sensitivity. Tell you what, let's get. So, just some final thoughts here. I put in another quote here, and we talk a lot about models. It's a big part of my sections. And again, models usually, they're not reality. They often don't, they're not able to explain everything and all the details, but they do allow you to understand complicated things in a framework. And so, the purpose of the model is not to fit the data. And often we don't really care exactly how well it fits the data. Really, it's how much is our understanding improved. And so it says, not to fit the data, but to sharpen the question. So usually, after you build a model, you have a few pieces that aren't fit by the model, and that tells you what's missing, what you should perhaps refine or improve on. This is showing that, remember in the Huxley model, it's two states. Cross bridges can be attached or attached, and I was saying, a model that's more consistent with their knowledge is that we have a detached state, and actually detached is detached or weakly bound. So a cross bridge may bind electrostatically, it's just not floating around. It may actually attach to a thin filament, but not really grab on. That's called a weakly bound state. And then we think it's kind of gloms on more strongly, something called this, which is pre-rotated. And then once it's more strongly bound, it can ratchet. And again, when it ratchets, it stretches the tail region. So we think this is sort of how it goes. And then this is a step where it detaches and it goes back to weakly bound. So, again, the Huxley model is very simplistic. It was very early. He didn't understand this level of detail about mice and attaching and rotating. So, let's skip this. This actually showed up in your homework. This signal here, for instance, is a dash pot. So, this is showing, something that's, if you didn't notice, but I'll just sort of clue you in, when the active force goes up, say it turns on in a step of 200 millinewtons, the dash pot's going to initially just balance it. So if that goes up by 200, this goes down by 200. Now it's only there for an instant because basically, as sort of things readjust and the dash pot goes away, steady state, the dash pot contributes nothing. So, and then this was something also you saw in the homework. And this is really sort of, if you were to turn on the active element, not in a square wave, which is where your homework, most of the homework problems are around, and what you will be responsible to solve on, let's say, a midterm, is a square wave. But if one was to draw something more realistic, it would go like this. It would go up quickly and then decay away as sort of an exponential. And then what you see is shortening becomes, this is sort of the length of the elastic, this is the series and parallel element. You can see the change is sort of like a twitch. The force now becomes like a twitch. So you see the peak of force actually happens as the active element is coming down in this kind of signal. And so just in the first lecture there was this setup, which is kind of complicated and sort of, it's intuitively difficult. But what this was supposed to show was that if you stimulate the muscle, in this case, it internally tries to shorten, but it can't fully shorten until you release that catch. And when you release that catch, later and later you get different transients. So at the earliest one you get this, later you get this, later and later and later. And just this point here at peak force is where there's no more shortening at the sort of in the internal element. In this case, it was a lucky case that we assumed, I lost my pointer here. What we did was we assumed that when we stimulated the active element shortened to the extent that the parallel element goes away, the parallel elastic element, and then basically at the peak of the force is where you get to see what the active element is doing, because at that point velocity is shortening to 0. And as I said, you could do this repetitively. And now what you can see is this dash is sort of the active element. So what we're going to kind of wrap up here is that when this active element turns on, it's sort of like the calcium turning on the thin filament. It turns on the thin filament and gives the ability for cross bridges to bind, but the cross bridges tend to be slower. And so, you know, even though it may activate quickly and begin to turn off, the cross bridge attachments are much slower. So the peak here of the force occurs after the peak of the calcium transient. Just 3 more quick slides. The other point here is I got this out of the paper just a year or 2 ago, and this is showing a model of muscle. And just the point here again is that, you know, what you're learning actually, people still use this today with series and parallel elements. This is a contractile element. So again, you can take what you learn and apply it. Last thing is, in the year 2000, I was invited to teach this course by David Yu. This is not to make anybody sad, but unfortunately David left us last year. But you know, I think unfortunately someone like Huxley, who I think really was going to be an influence on science, you know, David really did spectacular work. And so, like I say, 57 was the year for classics. So. And then this is who I work with now, which is Peter Mattoon, who's now at Loyola University. Turns out he also was born in 57. Anyway, I'll wrap up there. I'm going to hold a review session at 5 p.m. Is there any Mergenthaler? Do you remember?
111.
Yeah, okay. So I'll see everybody there. I'm not going to lecture again. You guys come with questions. If you don't have questions, I'm going to put things up on the board and make you solve them. But I'm not going to stand here and lecture again. So, okay, hope to see you then. Thanks, everyone.